So in this lecture, we're going to be discussing our analytical plan that we are going to be putting in place in our research proposal for our particular study. We're going to talk about descriptive biostatistics in this lecture. In the next lecture, we will talk about comparative biostatistics. And descriptive just means that you're describing a population. Comparative means that you're comparing populations. So our objectives for this lecture are to just discuss an overview of the analytic plans for the different study designs. We'll talk about measures of central tendency. We'll look at some graphical representations of data and how to interpret those. We'll talk a little bit about standard deviation and then some statistical honesty. So as we get started, we're just going to define biostatistics just so we're all on the same page. So biostatistics is the science of analyzing data and interpreting the results so that they can be applied to solving problems related to biology, health, environmental science, or related fields. And we're going to talk about the types of analysis that we can do. Right now, we'll talk about univariate analysis. There's also bivariate analysis and multivariate analysis. Beginning with univariate analysis, we're just going to take apart the word univariate. So uni means one, right? Unicycle is a one-wheeled cycle. So univariate means one variable analysis. So this type of analysis will be describing one variable in a data set using simple statistics like counts, how many people said this, how many people said that, proportions, so that would be percentages, and averages. So univariate analysis would be used in studies that have no comparison group. And remember when we talked about our different study designs, there were a couple of study designs that did not have a comparison group, and those were the case series or the case study and the cross-sectional study. So for those types of studies, univariate analysis would be appropriate, and they just describe the key variables that you're studying in your study. We can move on to bivariate analysis. So if univariate analysis is descriptive biostatistics, bivariate analysis by definition then would be comparative, because bivariate, if you take apart the word, bi meaning two, bicycle is a cycle with two wheels, variate, two variables, two variable analysis. We will be using rate ratios to compare populations, odds ratios to compare populations, and other comparative statistical tests to examine the associations between the two variables. And remember, we are studying associations. Analysis has to include both the descriptions of the key variables and the statistics that compare the groups. So the descriptions of the key variables would give us univariate analysis, right? If we have two different groups, we have to describe each of those groups. And then our statistics can compare those two groups. And when we talked about our different case studies, there were some that had comparison groups. Those were the case control study, the cohort study, also the experimental study. Had more than one variable, two variables. So the case control study, let's look at that first. We had two groups, right? Our cases and our controls. And what are we doing? We are comparing the odds of exposure between the two groups with the disease or outcome and without the disease or outcome. In a cohort study, we're looking at disease incidence, and we want to see who develops this disease. So we have this whole cohort, and we're following them through time, and which people actually do develop the disease. So then they kind of separate themselves into groups, people who have the disease or outcome, and people who do not. And then we can compare those using bivariate analysis or comparative statistics. There's also something called multivariable analysis, and that starts getting really kind of complicated because you are looking at multiple variables. What affects what in all these different relationships. We use things like multiple regression models, and they get really advanced, and you want to start using computers at that time. But I just want you to be aware, we're not going to be using that at all, but I want you to be aware that that exists. And so here is just a an encapsulation of the analytic plan and the study design that goes along with it, right? So if you want to describe your study population or your variable, you are using univariate analysis. And for the case series and the cross-sectional study, this is enough. Just describing what's going on with numbers, right? Your univariate analysis. When we get to our case control study, our cohort study, our experimental study, we're going to be comparing groups. So in that case, univariate analysis is not enough, and we have to take it a step further to compare our groups, and we can be using bivariate analysis, comparing two variables. We can also make it really complicated, but we're not going to. So we're going to talk about measures of central tendency. So for numeric variables, central tendency refers to various types of averages, right? When you have a group of people, you're going to have people of different heights. Most of the people are going to fall into the average height range when you have a high enough sample size. You're going to have some people who are taller and some people who are shorter, but there is a central tendency in human height to be around the same. So we have measurements of these average variables. We have the mean, we have the median, and we have the mode. And I know you guys have talked about this before, but let's just go through it real quick. So the mean is basically the average, right? You add up all your values and then you divide it by the number of values that you have. The median is the middle value, and then the mode is the most frequently occurring value in a data set. So real quick, we'll just look at these measures of central tendency. These are our values that are reported by our participants, and we are going to first look at the mean of these values. So what we do is we take all of these values and we add them up, right? Add them all up. And then we're going to divide them by 6. So when we add them all up, we get a certain number. Then we divide them by 6 because there are 6 values. 1, 2, 3, 4, 5, 6. And you get the answer. Your mean or your average is 39.5. We are going to look at our median. And so our median is going to be the middle value, right? So these are the values that were reported by our participants. So do we just look at 30 and 50 and find the median value? Well, no. First, we have to put our values in order from least to most, or, just put them in order. So when we put them in order, we get 25, 30, 30, 40, 50, and 62. We have 6 values, so we want the middle value. Well, there is no actual middle value because we have an even number of values. So what we do is we find our 2 values that are in the middle, and we find the exact halfway point between them, which is 35. Finally, we have our mode, and that's the value that appears most often in your data set. And so when we look at our data set, we see, 30 appears twice, and no other number appears twice. So our mode would be 30. There are different types of graphical representations of data that we can use. So this is something called a histogram. This is not a bar chart. This is a histogram. A histogram is something that we use with continuous data, numerical data usually, data that continues along in order. We can look at people's heights, right, from the least tall to the most tall, and we can measure them in inches and feet, centimeters, whatever you want to measure them in, and they go in order. That is called continuous data. So a histogram is a graphical representation of continuous data. So each bar is the same width, and there really shouldn't be any gaps in between these bars in the middle. And you'll see that, if you have a high enough population, sometimes they kind of organize themselves into a bell curve, which we'll talk about in a bit. But this is a histogram. There's also something called a box plot or a box and whisker plot. And so this just kind of gives you an overall representation that starts with the median. We find the median, and that is the midpoint in our data. And then, so in our case, the midpoint in our data is the value of 30. And then you want to divide the whole plot into quartiles. So you divide it into fourths, and you find your 25th percentile here and your 75th percentile here. This would be your 0 percentile to your 25th percentile. This would be your 75th percentile to your 100th percentile. The range of normal values that you have would be between 25 and 35. And then sometimes in data you have outliers that just kind of hang out there. But this kind of just is a graphic representation of all of your data except for the outliers. This is 100% of your data right here. This is the bottom fourth of your data. This is the second fourth of your data. This would be the third fourth of your data, and this is 4/4 of your data. Just a different way to interpret and display data. So a bar chart then is not a histogram. We already talked about that. A histogram is a representation of continuous data. A bar chart is going to be categorical data, and we'll talk about what that means in 1 second. These are equal width rectangles, right? With the lengths that are proportional to the values they represent, okay? What does that mean? Well, why don't we just look at an example? So this is categorical data, and this is a bar chart. So these are all different categories of responses. There is no particular set order for these responses to go into. They're not like numerical. They're not continuous. They're categorical. Let's say I'm taking a survey of what's your favorite pet. Here's cats, here's dogs, here's gerbils, here's fish, and here's birds, okay? You can put those responses in any particular order without it affecting your data because there is no inherent order to those categorical responses. And here is the count of the number of people who said that, what did I say? Cats were their favorite animal right there. So that is a bar chart, categorical data. We're also going to look at our pie chart. So that's the circle. So the circle represents 100% of your population, and then each slice is a percentage who answered whatever that categorical data point was, right? So we've seen these a million times. This is the population of students in our class. This is the number who prefer cats. This is the number who prefer dogs. This is the number who prefer birds. I don't remember, but that's representation of the same data in pie chart form. But you would do percentages here and not counts. So then, when we looked at our histogram, which represented continuous data, we saw that this continuous data fell into kind of a bell-shaped curve, right? And if you have enough human beings in your sample size and you measure their heights, they will fall into a bell-shaped curve, with most people being about average height. And then you'll have the taller people and the shorter people on the sides, right? This is called a normal distribution or a Gaussian distribution. Like I said, it has a bell-shaped curve with one peak in the middle. We can describe the shape of that curve. Yeah, it's bell-shaped, but sometimes it's a skinny, tall bell shape. Sometimes it's a flat bell shape. So we can describe that with something called kurtosis, and there are specific words that we can use, which we'll look at in a moment. There's also skewness, and this describes how asymmetrical a nearly normal distribution is, right? So it can be a normal distribution, and then it could be like leaning off to the side. So here is our examples of kurtosis. We have our normal curve right here, normal bell curve. Great. This one is really skinny and tall, meaning that most of the answers to the, question of how tall are you may fall really close to the average. Whereas where we have a platycurtive curve, we have more of a wide distribution of heights of human beings. Okay, so these are just words used to describe the normal curve. When we talk about skew, we talk about which direction our data has skewed off on, right? So this would be a left skewed curve, and this would be a right skewed curve. There's also, you can have a bimodal distribution where you have two modes, and then there's also something called a uniform distribution. So we can talk about standard deviation with our bell-shaped curve, and there are percentages that go along with our bell-shaped curve of where we can find our responses or our heights of human beings. We can say that 68% of our responses fall within one standard deviation above or below the mean. We can say when we talk about two standard deviations above or below the mean, we're talking about 95% of our responses. And then we have a number of 99.7%, and those are within three standard deviations above or below the mean. So this is what that would look like. This would be our normal curve. And we would find that 68% of individuals fall within one standard deviation of our sample mean. And that's all that means. So if we have our tallness of human beings, we have our tall people here, our shorter people here, our average or our mean is here, we would say that 68% of all the individuals that we had, if we had a large enough sample size, would fall within one standard deviation of the mean in terms of their height. 95% of all individuals would fall within two standard deviations of the mean, and 99.7% of all the individuals would fall within three standard deviations of the mean. So then we do have to touch on statistical honesty, because some people are not quite honest when they report their data. And reasons for this would be because of the prestige involved in publishing something that was considered really important in a highly respected scientific journal, and some people don't get the data that they want, and so they fudge it a little bit. This is not okay. Researchers are obligated to describe their data accurately and to correctly report the results of statistical tests. There is research misconduct, and three of the most serious forms are fabrication, which is the creation of fake data, falsification, which is the misrepresentation of results, and of course plagiarism, and that's the use of other people's ideas, words, or images without proper attribution. What we want is to discover the truth about a data set, right? So your statistical analysis is there just to discover the truth about your data set. It's not to creatively manipulate your analysis toward the result that you want. You want to report out your findings scientifically and as unbiased as possible, right? You want to discover the truth about the data set. It's also important that the researcher select the correct test for the question being asked and the variables being examined. And when it gets really complicated, there are statistical consultants. So that's always an option if you get really advanced. I just want to remind you, this is a really important graph to use. Think about your particular study design that you're going to be using for your proposal, your research proposal. Is it a case series? Is it a cross-sectional survey? You'll be using some univariate analysis, right? Is it a case control study, a cohort study, or an experimental study? You'll be using some bivariate analysis. So that's important to keep in mind. The bivariate analysis, of course, would be kind of like the incidence rate ratios, the odds ratios, the rate ratios. Those are the bivariate analyses. Okay. Next, we will talk about comparative statistics, and that will be in our next lecture.